Homopolymer tailing, it is a good technique for joining DNA from different sources. But nowadays it is not frequently used because of the availability of other techniques for joining DNA molecules. But still it has application in the cloning of cDNA or complementary DNA. cDNA or complementary DNA is a type of DNA that has been synthesized from the messenger RNA. मिसाल के तौर पर अगर हम यूकैरियोटिक सोर्स से या कोई हायर ऑर्गेनिज्म है उससे आरएनए को क्लोन करना चाहते हैं तो पहली कंडीशन ये है कि उसको डीएनए में कन्वर्ट कर दिया जाए. इसको डीएनए में कन्वर्ट किया जा सकता है जैसे कि हमने पहले डिस्कस किया है कि कुछ वायरसेस होते हैं लाइक रिट्रोवायरसेस, तो they can encode an enzyme which is called as reverse transcriptase, and this reverse transcriptase it can be used to synthesize the copy of the DNA which is called complementary or cDNA from the RNA molecule. How DNA can be synthesized from RNA? So like the DNA replication, here we also need a template as a starting point, and naturally RNAs they have a stretch of adenine residues at 3' end, like this stretch may be up to 200 adenine residues. So we can provide a primer of a few thymine residues that is complementary with adenine of the 3' end of the RNA, so that in the presence of reverse transcriptase, the first strand of the DNA it will be synthesized. जब DNA का first strand synthesize होता है तो messenger RNA को degrade कर दिया जाता है by using alkali. Alkali जो है वो messenger RNA को degrade कर देती है, लेकिन जो DNA है it is resistant to alkali. So after the synthesis of the first strand of the DNA, then second strand synthesis it is carried out by the use of DNA polymerase. So finally we will have two DNA strands, and then with this double-stranded DNA we can introduce homopolymer tailings, and as I explained you, and then it is ligated with the vector DNA, and then it can be introduced into a suitable host organism. Here we can explain the cloning of cDNA by the use of homopolymer tailing. So here we have the messenger RNA. As I told you that naturally this messenger RNA it has a stretch of 200 adenine residues at 3' end. So we can easily provide a primer that is complementary to the adenine residues, and with the help of reverse transcriptase it will mediate the synthesis of the first strand of the DNA, and the messenger RNA it can be hydrolyzed by the use of alkali. So this is first strand synthesis that is mediated by reverse transcriptase. So naturally the reverse transcriptase introduce hairpin-like structure at 3' end that can be used as a template to synthesize the second strand of the DNA, and this is mediated by DNA polymerase in the presence of all four types of dNTPs. So at other end of the DNA this hairpin loop-like structure it is there, and this loop-like structure it can be hydrolyzed by the use of some nucleases, so that one can get double-stranded cDNA. Then this cDNA it can be treated with terminal transferase here, so that it can introduce only, for example, cytosine homopolymer tails at 3' end. On the other hand, the vector DNA it can also be used to form homopolymer tails. So here the example is a plasmid which is called as PBR322. It is very good example of the plasmid vector that is frequently used in gene manipulation procedures. So this PBR322 vector, it has two selectable markers for antibiotics. One is tetracycline resistance, and second one, it is for ampicillin resistance, so that it can select the transformants when we will grow them on a medium that is supplemented with these two antibiotics. At the same time, suppose it has one restriction site for Ph.D1, so it can be digested with Ph.D1 so that we can get 3' single-stranded overhangs that can be treated with terminal transferase, but only in the presence of single type of nucleotide, which is here the guanine residues that are complementary with the cytosine of the cDNA. Now these two sources of DNAs from the vector or the cDNA, they are mixed together. So because of complementarity of the homopolymer tailing, they will join together. Cytosine will pair with guanine here. And these are a few gaps that will be created that may be bridged when we will transform it to a suitable host organism. So when we will introduce it to the host organism, the host repair machinery, it will bridge the gaps in the transformant DNA. So after transformation, the organisms or the host, it is grown on a medium that is supplemented with tetracycline or ampicillin. So only those transformants or recombinants will be able to grow on this medium that are having the genes for tetracycline or ampicillin resistance. So another advantage is that because now these transformants are recombinant DNA, it contains the site for Ph.D1, so the target cDNA, it can again be excised from the artificial recombinant DNA molecule. So finally, we can conclude that cutting and joining of DNA molecules that is mediated by different type of enzymes, it is a good tool, a very important tool in the field of gene manipulation.